Hello everyone, my name is Dukunimana Paul, student of Ines Ruhengeri. I'm studying a module called Applied Immunology. I have given assignment from question number 26 up to 30. So my presentation is all about antigen antibody complex. We are going to discuss about 5 questions. So stay with me because this video it is important. Question number 1, it is talking about what is the significance of monoclonal antibodies in immunoassay. When we say immunoassay, we are using antigen antibody complex reaction to detect antigen which is interest, or when you have antigen you can detect antibody by using this antigen antibody complex reaction. So when we say immunoassay are laboratory techniques that detect and quantify specific molecules such as antigen or antibody by using precisely antigen-antibody interaction. To mean that when you have a specific antigen, you can find antibodies of interest in any serum or any sample. So how? When your antibody is going to be monoclonal antibody, when is having if they have a specific to a single epitope of the antigen. So this antibody, they are having the part, this part is called, let me have a color. So this part here, it is called paratope. It is part of antibody which bind with epitope on the antigen. So if this antibody is a specific to single antigen, this antibody is going to be monoclonal antibody. So monoclonal antibody is having a huge significance in immunoassay because they are help in treatment. Like this one, it is called immunotoxin. Immunotoxin, it is a molecule which composed by antibody and toxin. So this is antibody, usually is monoclonal antibody. It is coupled with antigen, so the toxin. So this antibody, it is injected, and because it is specific to, for example, to tumor receptor, and then go and bind with the cancer cells. Then after binding there, this toxin can be released and then go to kill that cancer cells by inhibiting protein synthesis. So it means that when we say this question, when we come back on this question number 26, when we talk about the significance of monoclonal antibodies, so they are highly specific, as we have seen that it binds a single epitope of the antigen. So it ensures precise detection. So it means that by having monoclonal antibody, we can find a specific antigen of interest. So we are having also consistency. So where there is a uniform quality improving reproducibility across the test. So these monoclonal antibody are important in the immunoassay, where is going to be unique quality improving. So another importance of this monoclonal antibody in immunoassay, it is enhanced sensitivity to detect very low level of antigen accuracy. So to mean that in the sample, if we are having a very few antigen, we can use this monoclonal antibody, and then this very lower level of antigen you can detect by using this monoclonal antibody because it is specific. So monoclonal antibody also can be used in ELISA, can be used in lateral flow SC, and also can be used in the diagnostic platform, where like in the diagnostic platform you can say like CRP, ASTRO. Those tests, we are detecting a specific antigen or specific antibody, and then by having synthesized antigen or antibody, and when they make a complex, you can see visible agglutination or precipitation. So this monoclonal antibody, you are understanding that very important, because you can use this one, we can use in the CRP test. We can use this one in the ASILO, where we are going to check agglutination. So they are also used in therapeutic use, where is enabled to target immunoassay based on the cancer and disease treatment. So this is what I was talking about. It is what I was talking about here. in the what is significance of the monoclonal antibodies. So here, this is immunotoxin. This immunotoxin is helping us in the cancer cell treatment, where this monoclonal antibody go and bind with the specific receptor on the cancer cells, and then this toxin which is bounded on it is going to help us to inhibit protein synthesis of that cancerous cell. So let me continue on question number 28, talking about how does immunoprecipitation work and what is its application. So when we talk about immunoprecipitation, is a technique of precipitating a protein or antigen out of the solution by using antibody that is specifically bind to the particular protein. So to mean immunoprecipitation, it is a precipitation technique. We are going to use antibody or antigen, and then we are allowed to form antigen-antibody complex. So, and then we will put some more reagent like a protein to make this precipitation. Now, after precipitation, we can do extraction. Now here, by help of this example, this is how immunoprecipitation works. So this is the sample where we are targeting antigen or protein. So this sample, we are going to put the antibody. So let me have some marker. So we are going. This is an antibody. So this antibody we are going to mix with this sample. In the sample, as you are seeing, there is a protein of interest. So this protein of interest is going to react with this specific antibody, because we know that this specific antibody is specific to this antigen. They are going to bind and form the complex. So this complex, let we assume that it is formed because antigen is there. We are going to put add protein beads and then incubate. So this protein, what it's going to do is they are going to make this complex to be visible, they are going to make it precipitation. So this precipitation, now it means our antigen antibody complex is precipitated. Now we are going to wash and erode. So after washing, the protein which is a protein of not interest are going to wash away. We are going to remain with only protein of the interest. See, this protein here, it is not protein of interest. Now we see after washing is not being appeared. Now finally is the detection. You are seeing here it is a gel electrophoresis, and then this protein are going to separate according to their sizes. So small size we remove a little bit, but those which are smaller size, they are going to move far away. So by using this immunoprecipitation, you can having this sample, and then by help of antibodies, we are going to form a complex. After forming a complex, we are going to have this protein which is bound with the antibodies, and now we can have only protein, and then we can go to electrophoresis, gel electrophoresis, and separate them and then analyze them. So let we have a look on the steps how to do it. So firstly, it is antibody antigen binding. So as we have seen that in the sample, we're going to add the antibodies now to form complex. So this complex in this container we are going to add insoluble support. We have seen that it is a protein AG breed, which is going to make precipitation and then become insoluble. So after this, now we're going to do separation, where we're going to use centrifugation or magnetic separation. And then after we wash, and then we remain with the proteins as you have seen here. We are going to wash, and then we are going to remain with the protein, and then we analyze them. So in the analysis, isolated the protein is analyzed by using Western blotting, by using ELISA, mass spectrometry to identify and then study its interaction. So also in this question, we have been asked the application of this immunoprecipitation. So application of this, it's not quite many, but it is not complicated in this way, because in this immunoprecipitation, our target is to detect the protein or antigen of interest. So means application of immunoprecipitation technique is to isolate and detect protein of interest as the procedure says. So there is enrichment of the raw amount of the protein. So to mean that by using this immunoprecipitation, you can have, if in the sample there is raw amount of the protein, you can detect by using this immunoprecipitation. So there is also to study protein interaction and protein complex. So means in the sample, if there is many proteins, you can separate them by immunoprecipitation, and then you can study it, because now by Western blotting or gel electrophoresis, now protein is now being detected and now separated. So you can identify an unknown protein in the protein complex. So to mean that because you are having a specific the monoclonal antibody which is specific to that antigen, so you can have it means you can synthesize an antibody, and then this antibody can bind this unknown protein. Now you can study this. You can study this protein. So this one also you can verify protein expression. You can verify protein expression in a specific tissue. So it means that anything related by detecting protein, you can use this immunoprecipitation technique to detect it. So this is what's about on this question of immunoprecipitation, which is technique that use antigen antibody complex to detect a specific antigen. So let we have look on question number 28. So this question number 28, it is talking about what is immunocytochemistry. So in this question, there is two words. There is immunocytochemistry. So when we talk about immuno, it's all about immune system, it's all about antibody. When we say cyto, we are dealing with cells. So now this is a technique that assessing the presence of the specific protein or antigen in the cell by using the rapid antibodies. So to mean that we are in this immunocytochemistry, we are using the monoclonal antibody or rapid antibodies to detect a specific antigen. So this rapid, the reason to rapid these antibodies is to help in detection. So these antibodies is rapid by enzyme or fluorescence marker. So another question, apart from the what is immunocytochemistry, it is how this technique is performed. So I'm having a short summary here. This is the protocol of immunocytochemistry. So we take the cell or the sample and put on the slide. After putting on the slide, and then you do fixation, either heating or other way. So it means you are suspecting this sample to contain antigen. See, this red one is the one which is representing antigen in this the cells which contain the antigen. So what you are going to do, you're going to do permeabilization and blocking. So in the blocking, because we are going to put the primary antibody and the secondary antibody, you do blocking that we can prevent unnecessary binding, where the antibody can bind a specific protein. Now, after this, you're going to put the primary antibodies, which is rapid by enzyme, so that we can put substrate to have the color change of the reaction. So now finally, this is a microscopy. Within this microscopy, we can observe and see if there is complex, but this complex is not visible. You are seeing the intensity of the color change, which shows that the substrate is reacted by this enzyme, which is rapid on these antibodies. So let we see the procedures. Procedure number one, it is cell preparation. Fix cell on the slide and preserve structure. So this is the step number one, where the cells are captured and fixed on this slide. Step number two, it is a blocking by using the blocking solution like serum albumin. So this solution can help us to make the fixing and the empty space so that we can avoid the necessary binding. So we're going to put the primary antibody which is specific to the target of the protein in the cell. Now the secondary antibody, which is rapid antibody, is going to be added to detect the presence. Means they're going to be reacted with that primary antibody. This secondary antibody, it is wrapped by enzyme. So now at the level of visualization, we are going to add fluorescence or the enzyme. So this enzyme is going to, substrate on substrate. This substrate is going to come and now react with this enzyme. Now this enzyme substrate reaction, they're going to cause a color change. Now under the microscopy, we're going to observe is where we're going to observe the color change, and then we conclude that there is a presence of the specific protein in our sample. So this is all about immunocytochemistry. I hope we are going to, the application of it is to detect the specific protein in the cell, so by using antibodies. So to mean that antibody and antigen reaction is very, very important, because even is the one which is help in immunocytochemistry. So another question I have, it is how does indirect fluorescence antibody testing work? So this one, it is also called immunofluorescence technique. So this technique, it is indirect. When we're talking about indirect, it means that it is not one procedure. We're going to use means two antibodies to detect the specific antigen in the sample. So indirect immunofluorescence test, it is semi-quantitative. It means that it is giving the number, but which is not exact. They are sensitive, and then it is rapid test to detect a specific antibodies or antigen in the sample according to what you have. So to mean that if we are having antibody which is known, we're going to detect antigen is now unknown. So if the antigen is unknown, antibody is known, to mean that it is known, to mean that it is synthesized in the industry or manufacturer, and then they are used in the protocol. So without further ado, let's start. Let's continue. So this one, the immunofluorescence or indirect fluorescence antibody testing, it means that we are having the antigen in the slide. This antigen, we are going to add the primary antibodies. The primary antibody, if it found antigen to be there, they are going to be antigen-antibody reaction. To mean that paratope on antibody and epitope on antigen, they are going to react. So if they react, when we talk about indirect immunofluorescence, indirect to mean they are going to use two antibodies. Number one is not rubbered. Then we are going to apply secondary antibody, which is rubbered with the fluorochrome. This is the marker which will be help to color change or visible of the fluorescence in under fluorescence microscopy. So in this immunofluorescence technique, the right microscopy can't work. We are using immunofluorescence microscopy. So now let me see how the procedure are done. So antigen fixation, to mean that antigen is fixed on the slide. So this is step number one. Step number two, there is serum incubation. This serum incubation, their target, it is all about their target. It is the blocking at the patient serum antibody bind with the antigen if it is present. So to mean that also here there is a step of blocking, where we are going to put the serum albumin to block empty space to prevent if there is no blocking, this antibody can fix here to have false positive. So now after this, we're going to put after putting secondary antibodies, after putting primary antibodies, we're going to put secondary antibodies. These secondary antibodies is labeled by the marker. Maybe here it is fluorescence or fluorophore. These fluorophore are going to be detected under, are going to be visualized under fluorescence microscopy. So I hope this question number 29, it is clear and understandable, because it is called indirect fluorescence antibody testing or immunofluorescence, the technique. It is the semi-quantitative and sensitive and the rapid test to detect a specific antibody and the antigen if present in the sample. So to mean that this technology, it is helping us to detect antibody and the antigen in the sample. Now after this, we can conclude that there is a presence of the antibody or present of antibody according to what we were testing. So my last question, it is question number 30. in my presentation, it is asking what is flow cytometry and how it is used in immunology. So this technique also, it is very, very important because it is help us to analyze the cell regarding to granularity, their size. So it is important so to understand, like for example, those white blood cells, they are having CD markers like CD4, CD8, those surface protein marker, they are going to detect it by using flow cytometry. So flow cytometry, it is a lab technique that measure physical, chemical properties of the cell as they pass through laser beam. So by using this diagram, so this is the machine where the flow, how flow cytometry work. Here we put the sample here. Now this sample, for example, is containing the cells. So this cells is passing here, and now when this cell reach here, you see here there is a source of light. Laser light source means the laser beam is going to pass in this direction. So in this direction, cells, they are going to be detection of fluorescence emitted by stained cells. So if this stained cells is here, they are going to be a forward scatter and side scatter on these cells. So when we talk about the forward scatter, it means it measures size of the cells. So when we talk about side scatter, it measures a granularity of the cells. So in the summary, forward scatter of the right, it is measure size of the cell, while the scattered right, it is measuring a granularity of the cells. If the cell is having granules, means the forward, what is the volume of the cells, and so on and so forth. You know that they like lymphocyte, they are having 12 to 14 micrometer. So means this size of this lymphocyte is going to be measured by using this flow cytometry. Now we know that like neutrophils, they are having different granules. So these granules are detected by using this flow cytometry. So let me see how the procedure this is flow cytometry work. The principle: the cell passes through laser beam. After passing this laser beam, the light scatter and fluorescence provide data on the size, granularity, and the marker of that cell which is passed in that machine. So like fluorescence labeling, cells are stained with antibodies labeled with fluorescence dye. Now it is passed in that machine. Data collection. Now a measure means this machine is able to measure forward scatter to mean the size of the cell. Like as I said, if it is the lymphocyte, we say that having 12 to 14 micrometer. So to know this size it is because of this forward scatter. Side scatter means it measures a granularity. As I have said, the neutrophils, they have granules. To know those granules, it is better. They are read by help of this flow cytometry. And then there is also marker of the surface protein, like in those Hodgkin lymphoma, multiple myeloma. Those diseases are because of their lack of surface marker or the presence. So because of those surface marker, we can conclude the diagnosis. So by help of this immunocyto flow cytometry, we're going to have the marker of the cells and then help in the studying of the different diseases. So and now finally, a final step of this flow cytometry, it is data analysis. So analyze data by quantifying the cells expressing a specific marker. So means by having those marker, we can conclude that there is a problem regarding to our finding of the flow cytometry. So this is the end of my presentation. I'm thanking everyone which helped me to get, which are going to understand me to the end. Thank you. I'm Dukunimana Paul. Stay tuned. Next video are coming.